All right, it's very likely I'm going to be making some changes to the PowerPoints, by the way, or slideshows, just to make sure we're clear on that. So don't print off slideshow note-taking things way in advance, okay? Worst that happens is you'd have to cut parts out. So I'm going to be taking a butcher knife to the PowerPoints probably, because I just made too much details. Okay. So as we can see here, you see this equilibrium, and what does this equilibrium tell you with its arrow that's about the same? Quick. Stress. That equilibrium arrow being approximately the same means that the energies for these two behaviors, i.e. to bind to water versus binding to each other for these, this is the bases, right? We have the nitrogen with the hydrogen bonding to the carbonyl, right? These energies are actually pretty close. They're not exactly the same, but they're close enough that you're not going to see a large amount difference between the equilibria, and then you have to worry about other behaviors. So they're close enough, okay? So really what that means is that there actually is no real driving force to have the hydrogen bonding occur between the bases versus the water, okay? The real driving force here for that hydrogen bonding is actually that there is a driving force to match up versus not match up, okay? There's a big difference there. But there is not really much of a driving force to actually bond. What the real driving force typically for these assemblies is more towards the van der Waals forces within this. So you may have noticed that the bases are coming together like this, right? They're coming together with the hydrogen bonding happening in the middle. But then when we're twisting around and we're making this helix, what are we doing to those bases that are mostly organic compounds? They're getting stacked, aren't they? Okay? Now I know that you guys have seen this stupid picture that keeps getting put out, and ball and stick models, right? You guys are used to the ball and stick models. You see them everywhere. It's how you see everything, right? But is that how molecules look? Not at all, right? A bond is not a damn stick, right? A bond is an area electrons exist in. It has a considerable volume, doesn't it? Okay? And so if you take a space-filling model, this is more like what it looks like. And these would be the different orbitals around the different atoms, showing it where the electron density would be located. And what we can quickly see is that there is no hollow in the middle of this dang thing, right? You look at a ball and stick model, it looks mostly empty in the middle, doesn't it? No, this is like braided rope. It's pretty much solid, okay? There's not a whole lot non-electron filled space. Does that make sense when I say non-electron filled space? Yes, it's mostly empty space, but that's because the electrons are in those areas, right? So, does that make sense? So what you have here is you have your bases actually stacking on each other with slight turns each stack. Does that make sense? So each time it just slightly turns and stacks more. And then what this causes is it's going to cause a van der Waals contact and van der Waals forces. Now remember, these bases are not that small, are they? They're actually pretty good size, okay? And so you get a hydrophobic effect, and this actually is a large part of the energetic favorability for creating these double helixes, okay? And so the surface complementary occurs when the hydrogen bond donors align with the hydrogen bond acceptors and the nonpolar surfaces come together. This is why it arranges in the way it does. And it's energetically favorable to arrange that way. As we saw from the way back a few slides ago figure where we were talking about how they automatically assemble together actually quite quickly, right? Assuming we have all the ions and everything else we need. Okay? So the first law of thermodynamics is the total energy of a system and its surroundings is constant. The second law of thermodynamics, the total entropy of the system plus that of its surroundings always increases. And the entropy can decrease locally in the system if there is a corresponding increase in entropy in the surroundings. Another way of saying this is the bank account always has to be the same, but the tax man always takes his cut. That's a little colloquial. So what we're seeing here is this is the definition of entropy. And so the change in entropy of the surroundings has to be proportional to the heat, which is this enthalpy right here. We represent the enthalpy because it's under constant pressure. Transferred from the system to the surroundings. That's why this negative value is right here. And inversely proportional to the temperature. In this case, it has to be an absolute temperature, so Kelvin. Okay? Okay. By the way, this equation is actually quite interesting. What happens if you actually hit absolute zero? Stop moving. We get something even worse. It's undefined, isn't it? Goes to infinity. This term, this entropy going to the surroundings, would go to infinity at absolute zero. Everybody see that? Because you'd be dividing by zero, right? Yeah. And, absolute zero is this theoretical value, but there's actually some very good indications that we can't actually get to absolute zero in this universe. So, kind of an interesting thing. Anyways, so the total entropy change is given by this expression right here. So entropy change total would always equal the system entropy change plus the change in the entropy for the surroundings. Okay? That's actually just definitionally true due to how we define system and surroundings, right? What is a system and what is surroundings in chemistry? System's what we're looking at. We can define it as anything, right? Surroundings then are everything else that we're not looking at. Okay? So if we substitute one equation into the other, so the first equation into equation 2, this would actually give us this, which you guys are actually probably starting to look like something you know. Okay? Now please note that you probably thought you were done with entropy back in organic chemistry because basically nothing required entropy and we didn't care about it almost ever. But because it's a water system, what creates the melting effects of most compounds? How do you determine whether you're going to be a solid, liquid, or gas at any given temperature? Does temperature have any effect on enthalpy? The enthalpy term. Not directly.
Is that slash? Is that something like
Divide. Yes. Yes, this means divide. Because this is not using equation editor. Because equation editor breaks everywhere all the fucking time. Yeah. So you'll see that a lot where I'm just going to start using divide by, and I'll end up with terms on both sides of it. Yeah, unfortunately. Nope, it's division. Okay, so delta S total is going to equal the delta S system. Notice we have substituted this term into here. See that? First term. And for delta S surroundings. Okay? Okay, and so this is starting to look very familiar. You may recognize this from the delta G equation. Looks very similar, right? Yeah. And so what we can then do is we're going to go ahead and multiply over T. And what we see is we get negative T times delta S. This is why delta G is a negative term if it's spontaneous, by the way. Yeah. Weird, right? You didn't know that, did you? Yeah. Because entropy always has to increase for the universe. So delta G needs to be negative so that this term will be positive. Because this is delta G over here. Okay, and then that's the delta H of the system minus T, the temperature in Kelvin, times delta S of the system. Notice we have defined everything by the system. Why are we defining everything by the system? That was the goal of this fancy map. Can we measure everything in the universe? That's surroundings, isn't it? By definition, surroundings have to be everything but what we're looking at in the entire universe. So we measure everything on the system because that's what we can actually contain and look at, right? Yeah. And that's why we have this fancy equation right here. Okay. Minus T, the temperature in Kelvin, times delta S total for everything, including the universe, our system, surroundings, everything, is Gibbs free energy. Okay? Again, the reason spontaneous, if it's negative, Gibbs free energy is because that means we have a increase of entropy for the universe, don't we? Okay? And so again, what is the importance of this term, delta G? If delta G is 0, what does that mean? We're at equilibrium, aren't we? Yeah. We're not spontaneously going in either direction. Okay? Okay, if delta G is positive, what does that mean? Going from reactants to products would be not spontaneous, right? Non-spontaneous. Okay? Going the reverse direction would be spontaneous, exactly. And if it's negative, what does that mean? Reactants to products is spontaneous, right? Okay. Products to reactants would be non-spontaneous. That's when it's negative. Yeah. When this value is negative, going in the normal direction is spontaneous. And if you have trouble remembering delta G, if you remember what delta G is, which is the opposite sign of delta S, that actually is the easier way for you to remember. Because delta S total always has to increase, right?
So if it's at 0, then it's
Equilibrium. You can think of that as being it's spontaneous in both directions, or it's non-spontaneous in both directions.
And if it's negative, then it's going from
Reactants to products.
negative to reactants.
Spontaneously. And then positive is always going reactants to products spontaneously. Or another way of saying that means the same thing, is reactants to products is non-spontaneous. Okay, so if it is a positive delta G, that means it's going backwards spontaneously. Products to reactants. Going the forwards direction, the normal direction. That was positive. Yeah, it's positive. Yes. Not necessarily. Okay, let me put it this way. The conversion of CO2 to acid, you want that at your muscles, right? You want to be able to move the CO2 around at your muscles, don't you? But do you want it to be, do you want to be able to breathe the CO2 out later? Yes. Yeah. And so you need it to be spontaneous under one set of conditions and non-spontaneous under another set of conditions. Does that make sense? Or like O2, your hemoglobin transporting O2. It needs to spontaneously fix O2 to the hemoglobin at your lungs, right? And when it gets out into your limbs, what does it need to do? Spontaneously release it, right? Okay. So what most of your body is it's actually equilibriums that you're pressing on one side, depending on where we are in the body. Does that make sense? And what most of your biochemistry is going to be is it's an equilibrium that's getting pressed down by another equilibrium, that's getting activated by another equilibrium. Okay? And so we'll have these cascades of equilibria that sort of press down on one side of an equilibrium and cause it to be spontaneous in one direction. That kind of making sense? Yeah. And that's why we're sort of talking about this, is because, like, for example, what causes water to melt? The temperature, right? Why does the temperature cause water to melt? This term right here. When the temperature is low, this term is very small, right? When the temperature is high, this term is very large, right? And so that's the dictation of why water melts at a certain temperature, why anything melts at a certain temperature, right? Okay. And we're going to see this is actually related to concentrations as well, okay? Which you actually already know, but you have forgotten almost certainly. So, it's okay. Okay, so calculate the delta G if delta H is negative 93,000 joules per mole. Delta S is negative 198 joules per mole Kelvin. This Kelvin's on the bottom, by the way, with that mole. And T is 293 Kelvin. So we just insert each of these into their spot in the equation, right? i.e., delta S gets multiplied by the T here, and that's a negative value, isn't it? And then you take this value right here, you add that to that result. So you have two negatives adding together, and you'll get an overall negative value, won't you? One of the things you got to look out for if you ever need to do one of these problems is this sometimes ends up being kilojoules per mole instead of joules. This is almost always put in joules per mole Kelvin because this would be a very small value in kilojoules, right? And so you oftentimes have to do a unit conversion there, okay? Watch out for it. Okay, and so we go ahead and do the math. There's the equation. We substitute those in. We do the math. Notice here that this value right here is going to flip sign because we had a negative delta S value right here. See that? Oh, yes it is. You're right, exactly. Yeah, it is. Here, correct it for me on the thing. Come in and we'll get you bonus points, okay? If you have time. Okay, so the second law of thermodynamics. Second law states that for a process to take place, the entropy of the universe must increase, which is possible only if the delta S of the system is greater than the delta H of the system over T. Or T times the delta S of the system is greater than the delta H of the system. These are actually the same thing, we just multiplied it over, right? Okay. By the way, this is oftentimes colloquially known as you have to pay the piper. Okay? So in other words, entropy will increase if and only if delta G equals delta H of the system minus T times delta S of the system is less than 0. Keep in mind that's because of the signs, right? The negative signs in here. Okay? So biochemical reactions will occur only if the delta G is negative, which is only when the universe entropy increases. By the way, do you guys want me to define entropy? Because a lot of times people don't really. Okay. Do you guys know what a temperature is? Randomized kinetic energy, right? Okay. So what entropy is it's an increase in the number of possible states of the universe. Okay? It's not really randomness. That's not correct. Okay? It's just an increase in the number of possible states we could be in. Okay? Like if we had more desks in this room, there'll be more possible states that you guys could sit down in, right? Okay. Okay, so heat is going to be released in the formation of our double helix. This right here is the process of mixing. This will then be the reacting in the form of hydrogen bonding and van der Waals forces clinching down. Okay? And then we get this right here, which is our assembled double helices, right? Helices. Yes, helices. Excuse me. My Latin is very bad. So again, that spontaneous formation of our double helix is going to reduce the entropy of the DNA molecules. Can everybody see that okay? Like this increases the entropy, right? This reduces the entropy. We have half the possible options, don't we? Okay? Which is a pretty big decrease of entropy, isn't it? This appears to violate the second law of thermodynamics. So heat being released by the helix formation, however, is going to increase the entropy not of the system, but of the surroundings, right? And exactly. And the heat of the formation, when we're talking about entropy, entropy actually does involve heat. Okay? It's like a common misconception that they're unrelated terms, but they aren't. As you may have remembered in our previous work we looked at, that's delta S to the surroundings right there, isn't it? See that? That's just heat moving at a temperature. They're related terms, aren't they? Yeah. So yes, you're exactly right. We're releasing heat out into the universe, the surroundings. That increases the overall total entropy. And so that's what's giving us that negative delta G, and it spontaneously assembles. Okay. Keep in mind when we're talking about all these things, there's a difference between thermodynamics, which is what we're talking about right now, and kinetics. Okay? You can be spontaneous and very spontaneous, and yet not happen for hundreds of years. Okay? It's very spontaneous for your gasoline in your car to spontaneously ignite, right? Or if you happen to have an electric vehicle, an EV, when it's charged, it's actually spontaneous for the battery to explode. Okay? But kinetically, it happens very, very, very slowly. Okay? Which is why you don't get, poof. And that has to do with activation energy, right? Okay, so at the single molecular level, the outcome of a particular attempt is just not predictable. These processes are often referred to as excuse me. Let me get some water real quick, sorry. Stochastic, excuse me. Man. So this right here would be a solution of single-stranded DNA. We expose a solution of complementary single-stranded DNA for varying amounts of times, and we would then probe for a double helix formation. In single molecules, we could get maybe, maybe not. It's only when we get a bulk of molecules that we start getting probabilistic outcomes, right? So given exposure time of 2 milliseconds, the double helix formation is observed 9.9% of the experiments. So what does that mean? I mean, they're going to most of the time collide. And remember, these things have to collide at the right angle, right? They have to collide at the right side. They have to collide at the right speed. If they don't approach each other at the right speed, they could approach each other too fast and reflect off each other, right? And so we basically have, over the course of 2 milliseconds, whatever number of collisions that is, that gives us a result of about 9.9%. It's going to go. Okay? And that's what we're seeing here. Neat, huh? Anybody have any questions about this so far? So it's spontaneous to happen. If you give it enough time or enough attempts, it's going to happen probably, right? But it doesn't mean it's going to happen right away. All right. So these are the fluctuations observed prior to reaching a stable value. This is your percentage of showing double helix formation. And this is the number of repetitions. And so what we see is if we give it enough exposure time, enough repetitions, we're going to get a very stable transition it very heavily favors double-stranded formation, so the vast majority will assemble, right? But eventually we will, after a certain amount of time, kinetics will resolve to thermodynamics, right? And that's what we're seeing here. This is kinetics, how fast it happens, okay? And that has to do with the activation energy, not the thermodynamics, which one's more stable, okay? This part is the thermodynamics, isn't it? Specifically thermodynamics as related to equilibria, okay? So we're reaching equilibrium here. All right. Did that make sense? Okay. Okay. By the way, can you actually get to 0? Can you get to all of them are assembled? No. There's always going to be some breaking apart. There's going to be some that just never run into each other properly. And it happens, right? Yeah. So you're always going to end up with some sort of equilibrium that you're not ever completely 100%. So the acid-base reactions involving the addition or removal of a hydrogen H plus ion, these are the Arrhenius acid base, of course. We will also have Lewis acid base reactions, but they will never be mentioned as Lewis acid and base reactions because it's biochemistry. I know. I spent all the time teaching you, but you'll know, okay? You'll never mention that. The pH is going to be a measure of the H plus concentration. This is defined by pH negative log H plus. Hopefully all of you have that in the back of your heads and it's not an issue. This right here is the auto-dissociation of H2O. This constantly happens in solution. And so H2O automatically dissociates as H plus OH minus. This happens to about 10 to the negative 7th of these. And that's where the pH of 7 being neutral water comes from, in case you ever wonder. Okay. Does anybody have any concerns here? Did I lose anybody with any of that? Okay, please let me know if I ever lose anybody, because it's entirely possible. So remember that any equilibria, we just talked about an equilibria, has an equilibrium constant, right? That tells us the relative positions of these things. Positions meaning which form would have what concentration, okay? And so the equilibrium constant for this is referred to as Kw. That's the ion constant of water, okay? This is how we would set up the equilibrium constant for any reaction. We would put products, the concentration of the products, to the power of their coefficients, in this case 1s, divided by the concentration of the reactant. But something weird happens when we get stuff that doesn't have concentrations, like pure liquids or pure solids. What do they go to? They go to unity. Unity meaning 1. Okay? And so this H2O here, this is a pure liquid. It can't have a concentration of H2O within itself, can it? Like, what is the concentration of H2O dissolved by H2O? Unity, 1. Yeah, so we'll put 1 in here. That'll end up getting dropped. So the Kw of water can be defined as the Kw equals K times concentration of H2O. This can actually be simplified down, and this is actually effective to get something, as Kw equals the concentration of H plus times the concentration of OH minus. Okay? And that means typically you end up with a 10 to the negative 14 here. That's what that value is for normal water. They haven't done anything to, like put a bunch of iron in it or something. And that would mean that because these have to be equal, why do those have to be equal? In a neutral water, whatever you make of H plus here, you have to make the same amount of OH minus, right? Yeah. So in neutral water, I haven't put anything else in. These values are equal. So they end up being 10 to the negative 7th, 10 to the negative 7th. Gives you 10 to the negative 14. Why is the pH scale in water 0 to essentially 14? Huh? It's actually negative 1 to 14 point something, but close enough. Why is that pH scale, when we talked about general chemistry, that? Yeah. We're literally reacting this, aren't we? Yeah. So if we put something really, really acidic in here that's negative 15, it's just going to react all the OH minus out, and we'll be left with this, right? Yeah. And so those are the limits of H3O plus, the acidic form, and OH minus, the basic form. Anything past that just reacts completely, and we're back to water. Yeah. No difference. Okay, and so this is what I just mentioned to you. That's all this says. Kw, known value of 10 to the negative 14. If we calculate these out, what we find out is because these have to be equal unless we add something to it, the H plus equals the OH minus, which gives 10 to the negative 7 molarity. You may be wondering why the hell this would be important, but a lot of the energy production in your bodies is going to depend on this equilibrium and pushing this equilibrium one direction. Okay? Okay, so if H plus measures 10 to the negative 3 in a solution, what is the solution's pH? Yep, just 3. And all we do is just negative log of 10 to the negative 3. Does everybody know how to do logs in their head? Yeah, okay. Anybody that's going to be doing the MCATs, make sure you know how to do logs in your head, at least approximations. Okay? You don't necessarily have to be able to do them perfectly, but you need to be able to get approximations. So the acid-base reactions, these can disrupt the double helix, of course. So this, again, is the fraction of molecules in your double helix form. This would be 100%. This would be 0 down here. And as we can see, if we change the pH from 7 up to 11, what ends up happening is we actually get a curve that looks very similar to sort of an acid-base titration curve, doesn't it? See that? Yeah. And that's because that's basically what we're seeing. All right? At a certain point, our double helix is going to start acting as an acid, right? Yeah. And so what we're going to be doing is we're going to be changing the charges on it. This is going to cause it not to be able to associate together anymore. Okay, and so this is actually the acid that's being used here. And that's the guanine here is the one that typically deprotonates first. It's sort of your most acidic part of this, right? So again, the chemical basis of your denaturation is going to be the disruption of your base pairing because we're losing this proton, thus we can no longer hydrogen bond. It will also build up negative charge in the center where those bases are supposed to be paired, right? And that's going to disrupt that. This is an example of this. We have a pK of 9.7. If we look very closely here, we can see the middle here is somewhere around 9.7. See that? Okay. Anybody know why I can do that? Anybody remember the Henderson-Hasselbalch? pH equals pKa plus log of base over acid, I think it is. I always forget. You can do the Henderson-Hasselbalch in two different ways. Plus log of base over acid, minus log of acid over base. I believe. I might have gotten the plus or minus wrong. Yeah. Okay. Anybody have any questions about this so far? All right. So one of the ways to denaturize DNA is to just make it very basic. Okay? Hey, I wonder what one of the major methods of cleaning up surfaces and destroying viruses and bacteria and stuff is. Could it be using a cleaning solution that happens to be basic? Yeah. Using cleaning solutions that are basic, right? Why does that work? Because, well, number one, you probably are going to destroy their cell membranes, right? But the second part is that you also destroy their DNA. Okay, so right here is the proton dissociation. This would be for an acid. This is one of the methods you can represent an acid, which is HA, right? Which can represent the hydrogen being attached to an anion or the hydrogen still being on the acid. Okay? And so its equilibrium constant for any acid is denoted by this expression, which is the Ka value for this, the acid dissociation constant. This is going to equal the H plus coming off times the anion that's remaining, stuff left behind. You'll end up with a negative charge because you lost an H plus. And this whole expression is divided by HA, which is the remaining acid, okay? The stuff that did not dissociate. Okay. Why are all these numbers ones? Don't we have polyprotic acids? Dr. Clausen asking us things and expecting us to know things again. By definition, what is an acid-base reaction? It's losing a proton and donating a proton, right? One proton. Yeah. And so by definition, every acid-base reaction happens with one proton and can only have one proton, one anion being formed. Okay? And if you have a polyprotic acid, you would just have multiple stages of this. Okay. All right, so this is the pKa. P anything is what? Got it, negative log of, right? And so we have the negative log of Ka, don't we? Okay? And then we take the negative log of the H plus equals the negative log of this right here, when the pH is equal to the pKa. Notice that this is actually what I just showed you, isn't it? When I said it's like, this is around 9.7. Yeah. And the H plus has to equal then the H plus times the A minus over the acid right there. Okay. If we divide this across by H plus, that gives us 1 equals this. That would, if we then multiply that over, that would give us the acid that we have used up equals the acid we have remaining, doesn't it? Notice on this graph, if we go halfway down through this, what does that mean? Halfway would mean that half of our acid has been used up, hasn't it? Hey! pK equals pH, doesn't it? Yeah. Okay. So the N1 proton of guanine has a pKa of 9.7. When the pH is near or exceeds 9.7, your proton is going to be increasingly likely to be lost. Remember that these are not linear reactions, right? They're logarithmic. And so what does that mean? As we approach 9.7, we drop off drastically, okay? We go from 90, 80% is still double helix to whoosh, almost down to 0 within like 1 pH unit, right? Okay, so very fast. The base pairing will be disrupted. The helix will become denatured, meaning it unravels and is no longer going to form that helix shape, which scrunches it up a bit, right? Actually scrunches up quite a bit. The buffers are going to resist changes in pH of a solution. Buffers are going to be most effective at a pH that's near its pKa. Because again, as we start getting too far off of its pKa, we're going to have almost all of it consumed at that point, right? And typically this happens within about 1 or 2 pH units, okay? That you'll consume almost all your buffer and it's no longer particularly effective. Everybody remembers buffers fine, okay, right? Okay, so this is the primary buffer in most organic systems. This would be considered carbonic acid, okay? Essentially, it's the acid, well, it's the carbonic, it's the deprotonated carbonic acid, sorry. See that? We've lost the proton already. But carbonic acid, you have an enzyme that makes this from CO2, something like 10 million times a second, okay? This is actually how you transport the vast majority of CO2 in your body, right? Did anybody think you bound CO2 to your hemoglobin? Be honest. It's okay. Like, some books actually say that. Biology books. They'd be like, you transport CO2 back to your lungs with your hemoglobin. Just not true. It's carbonic acid. Okay, so this is a titration happening here. You add a known amounts of reagent to a solution, and then your reagent will react with, in this case, your base. So we're adding a known acid, and that's the drops, to our base, our buffer system here. And what happens here is, because this is a pretty effective buffer system, as we start reaching the pH equals pKa, we get a very gradual pH change. It really slows down. See that? Versus the DNA that went whoosh, right? The DNA went woo. And DNA is not a great buffer. Okay? Well, it's not that bad. So where is the sodium acetate's pKa? Yeah, right around 5. Yeah, that'd be 4.75 is what my slide says. Which is actually pretty close, if I remember correctly. All right, so this is the ionization reaction of a weak acid. This is given by this right here. This, using the double-sided arrow here for the equilibrium, actually is of real harm in this case, because a weak acid, by definition, the majority of it will not dissociate. So this side is favored fairly heavily in a weak acid, okay? Strong acids, the vast majority of it will dissociate. The equilibrium constant for this reaction is that same equilibrium constant we had talked about. And so if we take the logs of both sides, we'll get a relationship that you are very, very familiar with. And that is the Henderson-Hasselbalch relationship right here. And so again, this is the Henderson-Hasselbalch equation. I said plus for the base over acid, right? Woo, got it right. I think I did. Of course, you can always rearrange these. It's fine. You know, if you rearrange the terms in here, so you flip the denominator and numerator inside of a log, it flips the sign, right? Yeah. So if you ever need to do something like that, that's whatever. It's fine. So again, this actually shows you that relationship, that the buffers are most effective when you're within 1 or 2 pH units of their pKa. And remember, 1 or 2 pH units, how much of a difference is that in these terms? It's a log base 10, right? So 1 pH unit is a 10 times difference in here, right? That's a 10 to 1. Okay? 2 pH units would be a 100 to 1 difference. Yeah. Or a 10 to the second power difference. Okay, so this right here, the buffer functions, of course, best at its pKa value. And what you're seeing here is that we are slowly eating away at the amount of acid in these we are slowly eating away at the amount of the base, and we are creating more acid as we go along here. When we get up here, we've almost used up all of our base, and we essentially have only acid left. Okay? And that's why we suddenly get a massive drop-off, because we've essentially used up all of our base to neutralize the acid. So every drop of acid just lowers it to the pH that the acid is. Okay? All right, so physiological pH is typically near 7.4. Okay? That is the typical physiological pH. It stays pretty damn close to that, and it does not, you have real problems if you're deviating particularly far from this, okay? Though you do have pockets within your body that deviates massively, all right? And your body can survive a surprising amount of deviation. For example, you guys know of lactic acid buildup, right? In your muscles. And you know that you have HCl in your stomach, and that's like pH 1. Okay, so given the pK values shown, inorganic phosphate is going to exist in this state of equilibrium right here. We see that we're near 7.4, so what does that mean the primary form of this inorganic phosphate is in? So this is a polyprotic acid, right? Meaning we're going to be losing a proton and going to each of these. Does that make sense? Okay. At this pKa, we have 50% this, 50% that, right? Okay. And so we definitely don't have any of this form, right? Or essentially none. How about this form? Yeah, we have some of that, don't we? Yeah, it's going to be mostly towards this form. This is going to be our primary form of the phosphoric acid equilibria set, the species of phosphate. And then we have almost no phosphate, do we? Right here. Yeah. So we're somewhere over here, aren't we? Mostly towards this, but we have a little bit of that. Okay. So sequencing of the human genome was a landmark in human history, of course. It happened around the turn of the millennium. And at that time, we were like, we're going to have everything figured out. No. Because we don't know how it expresses the DNA, do we? Yeah. So the initial sequencing of a human genome, the sequence is determined. It has become much, much cheaper and faster now, though it is still not quite to the point where like you can just get it done willy-nilly for whatever reason. But you can get small snippets done, like 23andMe and stuff. And that's pretty cheap. Yeah. Full human genome sequencing is like tens of thousands of dollars sort of thing. Yeah. Insurance doesn't cover it. And we're not even sure if we want that to be something that people do. Look at all the 23andMe leaks and all that sort of stuff. Anyways. So identifying the sequence variation among individuals may be used to understand the predisposition to various diseases. This is something this is a very biochemistry take on this position, that there's an underlying assumption that this is a positive thing that we want to be happening. And, biochemistry course, cool. But realize there is very good reason some people say, hell no. Okay? And you might want to look that up if you're going to go into medical-facing careers and that sort of stuff. So this is the decreasing cost of DNA sequencing. And, okay, so you notice that essentially after 2000, because we had the big breakthrough right over here, that the cost started sliding down, and then we made huge breakthroughs. And the cost drastically decreased until it leveled off, and then it kind of does that over and over. See that? We make a big breakthrough, whoosh, levels off. Make a big breakthrough, whoosh, levels off. Okay? And so eventually this is going to become ridiculously cheap. Okay? It's not quite to the point where we can willy-nilly just get one done whenever we're like, I think I might have the precursors to thyroid disease. Okay? But it's getting pretty fucking close. Okay, this is the average difference between two people within one population or ethnic group. This is actually greater than the difference between the averages of two different populations or ethnic groups. The largest average difference in human beings is the male-female dichotomy. Okay? But racially wise, it's not a meaningful difference. And that's just the DNA. When we start talking about expression, phenotypical expression of that DNA, we have people within the same, any group you want to talk about, where one person will be literally 100 times stronger than the other person, or have 100 times the intelligence, or have 100 times the ability to do art or whatever, right? And that's called the Pareto effect, by the way, where if you take a factor of any group, for example, 100 individuals, 10 of those individuals will be produce 50% of whatever that is. Okay? So like publications, 10 of 100 produce 50% of the publications. And if you keep doing that, you'll just get that same effect over and over and over until you run out of whole individuals. So it really gets crazy like when you have 1 billion people. The top person in the world produces literally millions of times more stuff than the bottom person. Yeah. It's not even, it's ridiculous. And so this is the DNA expression, the phenotypical expression, the expression of the actual what happens is so much worse than this in terms of individuals. In terms of population groups, it's actually very little. Okay? Anybody have any questions about that? No? Okay, so this is the human migrations. This is actually supported by DNA sequence comparison. And so this is kind of fascinating how you can trace the human migrations through comparing the certain markers in the DNA. The mitochondria is usually the easiest one to deal with if you're looking at long term, but it loses a lot of the resolution, the short term changes, because mitochondria is conserved heavily. Okay, you guys know where you get your mitochondria, right? Your mother, and it's heavily conserved, very little evolution because you don't have two parents. Yeah. Okay. Let's talk about the human microbiome. Does everybody know what a microbiome is? It's the microorganisms that help to compose your overall what you consider yourself being, a human organism. Okay? Okay, so we have a complex microbial communities. In total, we call this the human microbiome. They live in and on the human bodies. They are evolving continuously, and we gain newly evolved microorganisms. Actually, we just gained one like, what was it, like 3 years ago or something? Yeah, one just evolved to clean like our pores. So there's a 10-fold more microbial cells than there are human cells in any given individual. So the vast majority of a human is not actually human DNA at all. Okay? And they are absolutely necessary for your life. So we aren't even, like what we think of as a human is not the human DNA only and the human stuff only. Okay? In fact, the other stuff is actually more important in some ways. In fact, there's actually really good indications that they determine your aging. Like if you take a microbiome from a young person and put it in an old person, they'll actually start reverse aging. Yeah. And that's what the rich do, by the way, right now. Have you heard of fecal transplants? That's what the ultra wealthy do. They get like really, really healthy people's poop and transplanted into a wealthy old person. That's what we're doing these days. It works. I don't know what to say. Anyways, microbiome. This appears to play a role in the maintenance of health and the occurrence of diseases. There's a lot of proof that one of the things that causes your susceptibility to disease is actually the fact of how many times you have your microbiome disrupted by full antimicrobial regimens. And so this is the like cutting edge, by the way, if you want to know. Okay? This is the science in the next 20 years that's going to be giving us 10 more years of life, 20 more years of life, whatever. Okay? And you really should learn this. And if any medical programs that you go into are not talking about this, there's a problem. Okay? This is the stuff. Anyways, here's again variable human microbiome. By the way, do you know what determines a fetus's immune system? The placental discharge. So if you have a C-section, the fetus is very likely to get sick unless you take the placental discharge and put it on its face. Weird, right? Because of the human microbiome. Okay, it was weird, but kind of gross, I guess, but sorry. Anyways, this is your host genome, host lifestyle, host pathobiology, host environment, transient community members, host immune system, host physiology, these all contribute to your core human microbiome. Core being the really important ones that dictate many things like your autoimmune diseases, your immune system, whether it will attack itself, whether it will identify cancer, whether it's going to cause excessive aging or cause less aging, etc. It's incredibly important. These are the major identified factors that affect that human microbiome. The most fundamental role of DNA is actually to encode the sequences of proteins. The proteins are built from 20 building blocks. These are called the amino acids. This is as opposed to the 4 building blocks that DNA are composed of, which are the different bases. Okay? Keep in mind that both of these things we're actually talking about the side chains to those polymers. Okay? So proteins will be spontaneously folded into elaborate three-dimensional structures. They're determined almost exclusively by their amino acid sequences. I took this from your book. This is actually kind of like an entry level, right? But in like when you look back at the end of this course, you're going to be going, "Oh, that was not right. That was kind of wrong." Okay. But yes, with it, if they have everything they need to do it, and they have all the machinery that would be in your cell, proteins will spontaneously fold. Okay? Without all that stuff, they can not spontaneously fold. Which is one of the reasons why DNA and genome did not tell us everything. One of the many reasons. This right here is a protein folding. And so right here we see an amino acid sequence, and it folds into this right here. We then have an amino acid sequence that's different right here. And what we can see is that this would then fold into a different structure. And that structure can be very, very, very massively different on almost every level of the structure. Okay? Okay. There is wiggle room, though. You do not have to perfectly have the sequence. Okay? And we'll talk about that a lot more way later on. We got some material to cover, guys. Okay, so comparative genomics is going to compare one organism's genome with the genomes of the other organisms. Sequencing studies can facilitate our understanding of health and disease, though keep in mind just looking at an individual's genome gives you only a very small amount of the picture. Okay? You have more of the human microbiome exists, and then you also have environmental factors, and then you also have lifestyle choices, right? And then you have DNA expression differences from those, okay? So a human genome provides you a very limited amount of information, but it is incredibly important because everything else builds off of it, okay? Does that make sense? Yeah. Okay, it can be used to understand other aspects of human cultures. For example, we can determine migrations. There's been a lot of interesting work done about that. And actually, we've had to rewrite several major portions of human history. You guys may not even realize. Yeah, so kind of interesting stuff. It will effectively demonstrates the unity of life. We're all made of those same building blocks here on Earth. Woo. They may have actually found alien genetic information, though, and a meteor crash site. So, anyways, that's an if. Maybe. Be pretty cool, though. The human microbiome does what? You guys go ahead and read and let me know. Appears to play a key role in diseases. Okay? Did you want me to go through and say why these don't work? No? Everybody's good? Okay. Okay, so this is the molecular clock hypothesis. The molecular clock hypothesis is, it suggests the extent of sequence difference between the same protein from two organisms could be used as a measure of the time that has passed since these organisms had a common ancestor. Does anybody know what a base assumption of this would be? What do you have to assume if this is correct? Well, yeah, you have to assume evolution. That's true. Yeah, you're exactly right. You also have to assume that these genetic changes in this evolution happen on fairly regular basis. That at the very least, it's a Gaussian type distribution or random changes, and not a direct, that something is causing an influence in it, and it's not entirely random. Okay? Yeah, I mean, that would be partially that you didn't have, the bigger one would be things like radiation. Yeah. Compounds, sources that would cause evolutions to happen rapidly. Yeah. Yeah. So active substances that would change these things. Anyways. Viruses. So viruses are comprised of a nucleic acid genome. And this really does seem to work okay. And under most circumstances, changes in DNA are pretty consistently, there's fluctuation, but it's pretty consistent timing. It's mostly random, right? Yeah. Remember, this is not actually evolution. This is changes in specific bases, okay? Which can lead to evolution over the long term. Does that make sense? Do you guys understand the difference between those? Okay. Like in aggregate, yeah, it would be evolution, but in individual, it'd just be mutation. And we're talking mutations that can happen. So viruses are going to be comprised of a nucleic acid genome, DNA, or ribonucleic acid, which is abbreviated RNA. We didn't have that yet in the chapter yet. Surrounded by a protein coating. Protein coating helps them usually penetrate through a cell. Ooh. For example, COVID. Oh, I shouldn't have mentioned it. Canceled. This has a spike protein. Yeah. These are not alive in that they cannot grow or reproduce on their own. Viruses always require a host organism. Okay? And not only require a host organism, but even if you gave them the nutrients that they need a whole cell. So they can't even survive just on the nutrients. That's how they're different than parasites or most diseases. Most diseases could. They infect specific cells and take advantage of cellular components to produce new virus particles. These have sequences that change over time, even slightly, every time the virus replicates. And so they mutate pretty quickly if you allow them. The comparison of viral sequences allows the timing of key evolution events to be estimated. The virus that caused severe acute respiratory syndrome, SARS, in 2002, 2004, this was actually termed SARS-CoV, and the virus that caused COVID-19, which is SARS-CoV-2. These actually diverged over periods of a few years from the viruses that were found in the horseshoe bats. And so we had actually a major shift between those two viruses. We can talk about COVID sometime if you guys want, but I'm going to try to not, because it just bogs us down too much. It ends up being a very long thing, and yeah, most people seem to be sick of it. So, and honestly, there's a lot of the science is just really fucking bad science. So. Anyways, biochemical knowledge is generated through the research efforts of human beings, individuals, and in teams at many different stages along a variety of career paths. Keep in mind that when we're talking about biological knowledge, especially involving human beings, we have people of very different pathways, thought processes, and training all contributing to this. So, for example, a medical journal has absolutely no reviewing of the data whatsoever in the peer review process. Did anybody go, oh shit? I hope you did, because that should be an oh shit moment, okay? And the volume of publications doubles every 72 days. in the biochemistry medical fields. Yeah. So this is sort of showing a sort of branching out of this. And so we have the college university systems that produce these individuals right here. These are then the institutions that are affecting that. And this is the fellowships between them. Fellowships are the programs that are run between the two. Okay? Okay, anyways, note, by the way, that we have grad school, master's, and PhD are already pretty different. They're at least somewhat related. There are at least people that are more on the scientific end of that and learn science, the scientific method specifically, and how to apply that. When we look at medical schools, pharmacy schools, veterinary schools, these people are actually closer to engineering. Okay? So they're more interested in using results, right? Which means that they're trained very, very hands-on practical to apply the current knowledge base. But they're not trained in things that the PhDs are trained in. PhDs are not trained in practical knowledge. Use the current knowledge base to treat people, right? Not at all. PhDs are instead trained to evaluate data, right? Yeah. And so you have very disparate individuals. It's the teaming up of these things that really creates the good science, okay? Okay, and anyways, this is some different advances through the efforts of the scientists. It's a research group working on, I forget what they worked on actually. Anyways, this is a research group right here. This is the sort of numbers you'd look at any major study for anything biochemistry related, honestly. 20 authors, 30 authors, very common. And putting out one publication every year would actually be amazing, okay? Versus me, 30 publications in like 6 years. Okay? I'm a spectroscopist, though some of mine are biochemistry related. But I only had to walk on for like a week or two, and then I got my name on. All right, and then she did some amazing work right here. This is Chelsea Pinnock, and it tells you what she did here, if you're ever curious. We'll go on to the next chapter. Does anybody have any questions? No?